welcome back to our monthly talks of the Foresight Molecular Machines Group. For those of you who don't know who I am, I'm Dr. James Arthur Cooper. I'm a lecturer in organic chemistry at the University of Reading in the UK. Today we have two talks, one from William Shih at Harvard and one from Lulu Qian at Caltech. Both, I think, are going to be based around DNA nanotechnology. That's at least what I know them for, but we'll see. So it'll be about 20 to 25 minutes on whatever they're going to present today. And then at the end, there'll be the forward-looking goals that we've had and asked the speakers to talk about for both presentations. This is for the speakers as well as everyone involved. So keep your questions coming in the chat function, Alison will prompt you to do that. And then what we've done recently, I think we'll keep doing it, is we'll have the speakers interact with the chat. That seems to work quite well and avoids issues with question comprehension if I ask them instead. And then we can also invite people to unmute themselves. We've had hands up before and stuff like that. So we'll play it by ear. It usually works fairly well, if that's okay. I don't know who's gonna, are you going first? Are you? Will, was that the suggestion? Or
I think it was
either way. I could go first if that's
yeah,
okay,
if you want to, that's fine. Then we'll do that.
Yeah, thanks.
Thank you. And then Alison's put a poll out for people to look at other things we want the group to focus on, things around, what is it on? Is it on networking? What types of meetings we want? Other things that people would like to talk about? Is this meeting time still appropriate? So please fill that in. The information is very useful for us. Do you have anything to add, Alison? So I'm trying to cover all the bases here.
No, very happy and excited for both of the presentations. And both of them, I should mention, are previous Foresight Feynman Prize winners. So super, super excited to have you both here.
Yeah, I was a terrible misstep on my part that I didn't mention that. So thanks for putting that in. So Will, the floor is yours. You can start whenever you're ready.
Okay, well, great. Thanks, James. And thanks, Alison, for hosting me for this exciting Foresight event. And I'd like to share with you guys today technology that we're very excited about that we call Crisscross Polymerization, that we see as we think it's a really important next big step for the field. And some applications that we're interested in are in the area of single molecule detection and identification. So within biotechnology, we have these really big 800-pound gorillas for detection identification, which I would say are ELISA, enzyme-linked immunosorbency assay, and mass spectrometry. But they're not very sensitive methods. Could we achieve the same kinds of sensitivity down to single molecule levels with DNA nanotechnology? And so I'd like to start off with DNA origami, which is one of the most important developments in the field of DNA nanotechnology. Many of you guys have heard of this. We have a long single strand of DNA bathed in a rich broth of oligonucleotide staple strands that are programmed with Watson-Crick complementarity to pinch together that long scaffold into these parallel loops after mixing everything together, heating up and cooling it down over maybe the course of an hour, maybe a few hours. And the most amazing thing about DNA origami to me is really how robust it is, that you basically either form an origami or you don't. And you get exactly the number of origamis that you add scaffold strands. Now, origami works really great for the size scale of maybe 10 to 50 nanometers, maybe 100 nanometers, but then currently it's not very good at extending to much larger scales. So if you have an application where you need much larger length scales, then we need additional methods, and that's what we think crisscross polymerization that I'll share with you will enable. Now, what kind of applications would you need much larger structures? And I'm going to share one which has to do with this idea of signal amplification. If you're trying to do ultra-sensitive detection of an analyte, that analyte is going to be too small to see, and you're going to want something that is not just thousands of times bigger than the analyte. You want something that might be millions, maybe billions, maybe trillions of times larger, much, much, much larger than an individual DNA origami scaffold. So we can pose in terms of the following challenge. Suppose that we want to be able to detect some biomarker representing an infectious disease. It could be genome of a pathogenic virus, down to really small concentrations, let's say 6 copies per milliliter. And this is kind of like trying to find a needle in a haystack, where in this case, if we do it to scale, we have a macro scale needle, and then the haystack is a mountain the size of the Matterhorn. So we can see that this is very, very challenging in order to find an analyte present in this mountain of garbage. So how can we do that? Well, we can think about writing two molecular programs in series. The first one is going to very reliably recognize whatever it is we're looking for and convert it into an artificial record that indicates, yes, we've found what we want. And we call that a nanobeam. Now, a nanobeam by itself is also too small to see. So we need to have a second molecular program that very reliably will convert each nanobeam into a micro scale beanstalk, or maybe even something larger than that. And the key here is that it's a process that needs to never, ever, ever get started unless it's initiated by a nanobeam. And so you might imagine using chemistry that we have trillions of these building block components that are poised and they're ready to polymerize into these large structures, and we've kinetically prevented them from doing so. And let's say that we've suppressed that unwanted nucleation down to 1 part in a million, which sounds pretty good. But if you have a trillion components, that means that you still have a million unwanted nucleation events. So what we really need is a method that can suppress unwanted nucleation down to, let's say, 1 part in a trillion or lower, which is very, very demanding. The solution that we're going after is using a kind of cooperativity, and the particular kind is we call crisscross ultracooperativity that I'm going to explain to you now. So here are the building blocks for the crisscross polymerization growth. We have these golden elements that are vertical, and then these blue elements that are horizontal. These are just, in this example, 63 nucleotide long pieces of DNA arranged into a helix. And they're programmed with sequence-specific interactions such that in this example, there's 12 unique golden vertical sequences and there's 12 unique blue horizontal sequences. And each golden sequence is programmed to bind with just half a base pair, so 5 or 6 nucleotide base pairs, to those 12 different blue slats, and vice versa. So each horizontal blue slat is programmed to bind site specifically to 12 of those golden slats in the arrangement shown here on the right. And the way to think about this in terms of the growth is kind of like beads on a string. So if we imagine that just plotting the midpoint of each of these elements that we call slats, they add in a line. But what's special about it is that each incoming slat is making contacts with neighbors that are very far away. So this golden one at the front here is making a contact with a horizontal one that's 1 position away, 3 positions away, 5 positions away, 7, 9, and 11 positions away. It's making a huge number of contacts to neighbors that are quite far away. And that's really the key to the magic of the system of suppressing spurious nucleation. And so what we do is we operate this self-assembly at a temperature where in this case, we need 6 bonds for stable attachment. So at this temperature, if we have a vertical slat that tries to come in this example, it can only make 5 bonds. And so at this temperature, it's going to fall off. But if we look at the horizontal binding site, there are 6 binding sites. The horizontal one is stable. And now look what it's done. It's created new binding sites for the subsequent vertical ones. So now when that green one tries to come in again, it now can make 6 bonds and the process can proceed. So now we can get to the crux of the matter. How does this very baroque, intricate, highly coordinated structure allow us to completely squash unwanted nucleation in the first place? So we can understand that from a reaction coordinate where we have growth on the x-axis and then the free energy on the y-axis. And we can see when we start the unwanted assembly that as we start to link things together, which is what we don't want to happen, that this is an unfavorable process because you need 6 bonds for stable attachment, which means if you're only making 1 bond, that's unfavorable. And so you're going up the energy landscape. As we start to add more and more of these elements to this growing assembly, we keep on making fewer than 6 bonds. So we keep on going up the energy landscape. And the key is that we can program the system with a custom number of these horizontal and vertical slats that need to come together in order to form a stable critical nucleus. And it turns out that the height of this barrier that we're trying to engineer a very tall barrier in terms of units of energy is linearly related to, let's say, the number of horizontal slats that we program in that critical nucleus. And really the bottom line is because we have complete control over how big we design this critical nucleus needs to be, that means we can make this energy barrier arbitrarily tall, such that basically nothing will ever happen unless we provide a seed. And so that gives us the opportunity now to have the system that thermodynamically wants to trigger polymerization, but just won't ever, unless we provide a seed, which would, in this application, would represent the nanobeam that indicates that a target has been identified. And so for this example, what we do is we have a DNA origami that organizes these single-stranded elements into this parallel arrangement that looks like a bunch of horizontal slats. So those stable strands of the origami are paying the entropy price of jamming those strands close together. And in that way, you can basically bypass this large energy barrier. And so we have these golden initiators that bind to the origami seed. And then as the process proceeds, then eventually you get a structure that just looks like the end of one of these crisscross ribbons. And you can get growth up to the right, as we can see here in this diagram. So on the left here is the model. We have a DNA origami. It's seeding the assembly of a ribbon. And then on the right is a negative stain transmission electron micrograph of the structure. The individual building blocks are too small to resolve by TEM, but we can see the aggregated structure as it grows. And we believe that it's about 1.5 nanometer extension per building block. The growth is relatively fast. So we've estimated a second order rate constant of up to 10 to the 6 per molar per second, which means basically it says that we can get addition of something like 1 unit per second at micromolar concentrations. So we can grow these structures. This is, I think, 600 nanometer scale, 100 nanometer scale bar, but we can grow these structures over 10 microns in length. So here's just some more images of different length ribbons. So it's quite a processive process. It's comparable in terms of the ability to extend to something like DNA polymerization. So DNA polymerase also will be able to extend things on the order of tens of thousands of additions. So it's comparable to that. We can also program these ribbons to undergo different twisting motions. We can also introduce little glues on the ends of these twisted ribbons so that they'll cinch together and form tubes. And we think this structural variable morphology might be useful for multiplexing, but just more general, we're just at the beginning of this process, and we think in the fullness of time, we'll be able to generate the same kinds of structural diversity with crisscross that we can with something like DNA origami. So far I've explained how it is that we can design a polymerization process that only will be initiated in the presence of a target. But how do we actually use that to recognize the target of interest? And that can be a tricky question, but we think we have a solution for one class of analytes, which is a long single-stranded DNA or long single-stranded RNA target. So for example, SARS-CoV-2 has a single-stranded RNA genome, and so that could be a target for recognition. What we do is we synthesize, in this example, 6 of these reader X slats that are basically like staple strands that will fold the target genome into a mini origami. And what happens is those green horizontal elements, they're held in high local concentration. Again, that entropy price is paid, and then that energy barrier can be hopped and the process can be off and running. And so we see here in the all-gross gel, this represents the target. In this case, it's the M13 single-stranded DNA. Here we can see where we add the building blocks that thermodynamically want to grow, but without the target, nothing happens. And it's only when we add both the target and the building blocks together that we get growth, just like we expect. So to conclude this first part of my presentation, we've come up with an architecture that we think basically combines the absolute scaffold dependence of DNA origami with the unbounded growth of DNA tiles. And so it's basically extending the advantages of DNA origami potentially to much, much larger length scales. And we think this is going to be very important for enzyme-free, low-cost, ultra-sensitive amplification for diagnostic type applications. I don't have time to talk to you, but we also think this could be interesting for nanofabrication of basically microstructures with nanoscale features, which currently is very difficult to control with self-assembly. So far, self-assembly, we're good at building things up to about 100 nanometers, but we think this is a breakthrough for extending that control to much larger length scales. And this is work that was really led by Dio Minev, Chris Wintersinger, Anastasia Ershova, who were graduate students in the lab when they did the work. Theo's now a postdoctoral fellow continuing in the lab. I'd like to thank the following sources for support. And I have a second story to tell you very briefly. So here's a completely different method that we've been designing in collaboration with Wesley Wong's lab at Boston Children's Hospital in order to characterize molecules based on their chemical composition. And we have the analogy here of a caliper that, let's say that there's some kind of target and we're trying to figure out its identity or figure out its structure. What if we had the ability to have a little nanoscale caliper that could make a bunch of pairwise measurements on the surface of the object? And if we collected enough of those measurements, we might be able to stick them into a computational prediction algorithm and then either figure out its identity or deduce its atomic resolution three-dimensional shape. And I don't have much time, so I'm not going to describe this in great detail, but just to give you a flavor of what it is we're trying to do here. So the first step is that whatever analyte we're interested in, we do have to chemically link DNA handles to chemically label elements of the sample. So let's say in this case, there's a peptide, and our goal is to figure out something about the sequence of the peptide so we could identify it from a database. So first step might be that we could attach one long sequence to, let's say, the amino terminus of the peptide. So that's a unique chemical feature that we can tag. And then we put a second DNA sequence on, let's say, the lysines. So that's the first step, or we could have maybe attached this long one to a cysteine. The second step is, now that we have things that we can grab onto, is we have a caliper, which in this example consists of two microscale beads that are held in place with a dual beam optical trap. And they're tethered to this long DNA strand that we call a caliper. And what the caliper is programmed to do is to grab the labeled analyte at two locations. So let's say it's grabbing the amino terminus with the bottom, and it's randomly grabbing one of the three lysines at the top. And what happens is when it engages one of those lysines, that loop is cinched into a smaller distance. And we can observe this if we can track the distance between these large microstructures, we'll observe a compaction of these two beads. And that's really our readout. And the key notion is that the larger the distance between the handles in the actual analyte, then the smaller the compaction of distance between the microbeads will be when we measure. So if we see a very large decrease in distance between the beads, then we know it's a very small distance. If we see a smaller compaction of the beads, then we know it's a longer distance. And it's actually quantitative. So what we can do is measure these over and over again, collect histograms of the lengths, and then we can deduce basically from the mean what the atomically precise distance is. And so just as some examples, so here we're looking at analytes of known structure. So we're just trying to calibrate here. So what we do is the analytes are just some number of thymidines. So it's a well understood polymer with a known length. And then what we do is we sample the actual measurements that represents these histograms. And then we collect the means of each of these distributions. And then one interesting thing is when we plot the means of these distributions on the y-axis versus the number of designed thymidines in the actual analyte, we get this perfect fit with residuals down to basically below 0.2 angstroms, except for this regime with a very small analyte. And we think that's because we get steric collision with the elements of the caliper. And then we have this deviation with a very long element. And we think that's because the analyte is now competing with the length of that loop. So if we just make a longer loop, we would be able to get rid of this deviation. But the overall exciting message is that, yes, we can use this mechanical force spectroscopy in order to measure distances down to the atomic level. And we can repeat this with a protein analyte. So here is two different peptides where we put a unique cysteine here at the labeled in blue. So we put a long handle on that, and then the free amines, we put a second handle there. And so what happens is the caliper is always holding on to handle 0, and it's randomly sapping 1, 2, and 3. And so what happens if we measure this over and over and over again, then we see these 3 different peaks with these 3 different means. And importantly, we can see that the correspondence with the predicted values is very, very close, within a couple angstroms. And then likewise here, this is another peptide. We rendered it here as a helix, but we probably shouldn't have. We're applying enough force that we're actually denaturing this peptide. And it's the same concept that we make repeated interrogations between this blue cysteine and then 3 amines. And then we can resolve 3 different peaks in our histogram. And the values that we observe are within 2 angstroms of what we predicted. And so what that says is that if we know exactly the distance between the amino terminus and these different residues, then we can convert that into a number of residues, since we know from chemistry that the distance of the length of a single amino acid is something like 4 angstroms at full extension. And so in that way, we can get a sequence fingerprint of the species and then use that to deduce its identity by comparison with a sequence database. So to conclude this second piece of technology from our group, we basically have designed a DNA nanocaliper that in this case is going to be operated with optical tweezers. This is work led by Prakash Shrestha in Wesley Wong's lab. And we were really excited because we demonstrated that we could get sub-angstrom precision for a couple of analytes that we looked at. The next steps are to get the precision even better. We think it should be possible to resolve sub-angstrom measurements from just a single measurement instead of having to make lots of measurements and then collecting the mean. And secondly, maybe more important, the optical tweezers are very low throughput. So now we're actively trying to re-implement this in a highly parallel, let's say, flow-based assay. So can we make, instead of 1 measurement at a time, can we make millions of measurements at a time? And our long-term dream with this technology is not really to look at denatured peptides, but can we make these pairwise distance measurements on native macromolecules? So you can imagine the dream of if we could take a cell and then crack it open and spills out billions of these complexes, and we could have millions of our calipers on a glass slide, each one grabbing a different one of these complexes randomly. So we're sampling 1 million out of 1 billion. And each one of those is in parallel, making a bunch of pairwise measurements on the analyte and reporting back optically what distances they see. And then we can feed those distances. We can create basically a structural 3D envelope that tells us the shape of the structure. And then we can feed that into a prediction algorithm like Rosetta, and then deduce the high resolution structure of the complex, which I think would be incredibly important. So again, this is a collaboration with Wesley Wong's lab and work led by Prakash Shrestha, postdoctoral lab, along with other team members, Darren Yang, Andy Ward, Tommy Tomoff, Jim McDonald, and Bhavik Natwani. So here I included a couple slides on near-term applications for this technology, and then also medium-term. So for the first thing that I told you about with crisscross polymerization, we think we're close to being able to implement this in a practical way that could be very important for society to give us low cost, enzyme-free, so it'll be very robust, it'll avoid a cold chain, and ultra-sensitive diagnostics. So can we detect very, very low concentrations of the analytes and maybe even count molecule by molecule how many copies there are? The challenge is for us to, now that we've done proof of principle, to increase the speed of the operation of the device, because people don't want to wait around for a diagnostic, and then to expand the scope to a larger number of classes of analytes. So far we've just shown that we can do this for long single-stranded RNA and long single-stranded DNA. And that's an important class, but one would like to also be able to analyze smaller nucleic acids, proteins, small molecules, et cetera. And the second technology I told you about was these DNA nanoswitch calipers that we think should allow us to do single molecule identification and structure determination. So those of you who attended David Baker's talk last month, the question was, what's the big challenge? And he said, the big challenge is we need tools for molecular characterization. And we believe that these DNA nanoswitch calipers might be an important way to solve that puzzle. The challenge in the next few years, one challenge is to increase the resolution of our measurements. Can we get down to angstrom resolution from just a single measurement? And then just thinking about, well, if we look at these technologies going longer term, what are the challenges and potentials? As we'll hear from Lulu, there's a possibility that we can construct molecular computers and molecular robots that can take input from lots of these different sensors. So maybe they could look at 50 different biomarkers, and can we program these molecular robots to be able to recognize disease, integrating a large number of inputs. I think that's a very exciting area. And then for the DNA nanostructure calipers, I think an important frontier, like I said, is can we increase the throughput of these measurements? So we're hopeful that within the next 5 to 10 years, we'll be able to indeed increase our throughput to something like 1 million complexes that we look at simultaneously per low-cost microscope. And with that, I'll stop and take any questions that you guys might have. So if we look at the chat, so let's see. James Tour, at these levels of detection, are they not too sensitive for medicine? You can find a few molecules of all sorts of things. Yeah, I think there are a lot of analytes where you could argue that this is overkill, and so that's important. Even for things where it's overkill, I think it can be advantageous if you can do it very, very cheaply. That means you could take a sample and you could dilute it a lot if it's already pretty concentrated, and you can make a lot of measurements from a very small sample. So even then it could be useful. But then I think the bigger excitement for me is there are a lot of analytes that are rare. So if we think of viral infection, we always want to know at the earliest point when we've been infected. So I think there's always a demand to get more and more sensitivity. Let's see, so Tom Khalil, do you think this would be a good candidate for ARPA-H program? Improve the platform technologies for single molecule detection and demonstrate health applications where early diagnosis of low abundance molecules would be really valuable. Yeah, absolutely. I think that that these kinds of technologies, either from us or in combination with other people, if you think of something like serum, it's basically this treasure chest of information about our health that we cannot open at this moment. There's analytes that are just so dilute that all those other abundant molecules are just swamping out the signal. We have no technical way to get at that. But scientists and physicians have banked away thousands and thousands of patient samples, blood samples, into freezers, and they've written down information about what their phenotype is. And so they're just waiting for new technologies to be able to increase the sensitivity and pull out that information. So for sure, we're going to have a revolution in health based just on lower cost, higher sensitivity detection of different analytes. And yeah, ARPA-H would be a great way to invest in that. Tom Kellel, lots of discussion in Foresight and adjacent communities about the need for reform, greater experimentation with new models for both funding and organizing research. Do I have a view on this? What would you do if you're Eric Lander and wanted to propose a new model? For example, NIH's experimentation with the people, not the projects, Adam Marblestone's proposal for focused research organizations, more use of ARPA model, et cetera. Yeah, I do like the idea of investing in the person, and there are some programs, there are some opportunities already for that, but I think, yeah, increased investment in people as opposed to specific projects can be useful. I think that having a balanced portfolio of these larger organized consortium type funding opportunities is good, but it's important not to diminish the traditional investment in individual labs, because I think a lot of innovation comes out of agile small groups of people, and it's important to not do just one mode of funding to the exclusion of others. But I think it's a great topic and worthy of a lot more discussion. Stefan Borsley, how can the calipers cope with molecules that can undergo large amplitude conformational changes like lots of machines? Yeah, so one way that we could reconfigure the calipers is if we could construct the caliper where we have one DNA strand and let's say it binds to one element of the analyte, and then we could put the other part of the caliper as discontinuous. So it binds, let's say, if you have one part of the caliper binds the N terminus, one part binds the C terminus, then now you basically can open the structure the whole way. There's no more loop that limits the measurement you can make. And then the way you can make measurements is you have another T handle that comes out is randomly binding different parts of the molecule. So you're basically turning the molecule itself into the loop. And so now you're just limited by the total length of the molecule, and you wouldn't really want to make any measurements larger than that anyways. See, Tad, maybe I'll just try to get two more questions very quickly. When we get info on many low concentration molecules in serum, how will we learn what that means in terms of health? Lots of new info to interpret. Yes, lots of information to interpret, and yeah, lots of deep learning to be done to extract that information. I don't want to take up too much time, so I'll try to answer any other questions in the chat and turn it over to Lulu. How does that sound?
Yeah, I think that's thanks for that, Bart. That was a great talk, and yeah, it's also a good idea to keep things moving along. So that's fantastic. Thank you. So we'll move to our second speaker, which is Lulu. So we'll hopefully the screen sharing will work seamlessly, which it has. So the floor is yours. So thank you very much.
I have to first unmute myself. Now, can you all hear me? Okay, excellent. I'll get started. So today I will talk to you about a story on molecular pattern recognition in DNA-based artificial neural networks. So let's start with a fundamental question. What does it mean to be smart? Well, you might agree that recognizing a situation correctly is an important part of being smart, and it is a prior condition for making a good decision. For example, it is not necessary to run away from a cat and not a good idea to hug a tiger. And recognizing a situation correctly is also important in a molecular environment. For example, this nanorobot made of DNA can recognize a disease cell based on biomarkers on the cell surface and open up its barrel such that the antibody loaded inside the barrel can be given to the disease cell, but not to the normal cell. However, recognizing things can be tricky. Well, if the meerkats are not challenging enough for you, how about the dogs? Anyone has an answer? Five? I heard 5. There's actually 8. 8 of them. It's a checkerboard pattern. If you stare at it for long enough, you'll see it's a checkerboard pattern. Okay, so recognizing things can be even trickier in a molecular environment. For example, there are over 1,000 aroma compounds in coffee, and it takes a very complex olfactory neural circuit to tell the difference between a good and not so good coffee. Inspired by how biological neural networks in the brain are particularly good at recognizing things, there are mathematical models. They have been developed to create artificial neural networks that can perform sophisticated pattern recognition tasks. And a particularly desirable property of neural network computation is that the same network could be easily reprogrammed to perform different pattern recognition tasks. So it has been hypothesized for about 30 years that neural network computation occurs not only at the cellular level in the brain, but also at the molecular level within individual cells. For example, genetic regulatory networks and receptor ligand interactions in cell signaling pathways could both be performing some type of neural network-like computation. So one of the simplest neural network models is the linear threshold function, where the output is 1 if the weighted sum of all inputs exceeds the threshold. And 10 years ago, working with Eric Winfrey and Shuki Brook, we developed a DNA implementation of the linear threshold function and built a 4-neuron Hopfield associative memory that can recall a 4-bit molecular pattern based on incomplete information. And so if you have 1 or 2 clues encoded in short piece of DNA strands, they were sufficient for a test tube of DNA molecules to figure out which of the 4 remembered 4-bit patterns is most compatible with the clues. So obviously, that was not smart enough for many tasks that you might want to program a molecular machine to do. So a question that motivated my lab's research is how much intelligence can be programmed into artificial molecular machines? When Kevin Cherry, a graduate student, joined my lab, he was determined to make the DNA neural networks much smarter. So he successfully scaled up DNA neural networks from recognizing 4-bit to 100-bit molecular patterns. And different from the linear threshold function, in Kevin's work, we implemented a simple competitive neural network model called winner-take-all. Instead of each neuron having its own threshold for turning on, all neurons have to compete with each other. And the only one that turns on is the one with the largest weighted sum of inputs. And this model is inspired by the lateral inhibition and competition observed among biological neurons in the brain as well. And one of the most important advantages of winner-take-all circuits is that the negative weights are not necessary here, because only the relative values matter in selecting a winner. This leads to simpler molecular implementation that requires fewer molecules. Now let's take a closer look at this winner-take-all function. So for a number of inputs that are real values, a binary output is 1 if and only if the corresponding input is the largest, which means the output indicates who the winner is. And adding a layer that computes the weighted sum of a number of inputs before the winner-take-all layer allows pattern recognition. For example, let's consider a circuit that is designed to distinguish a letter L from T. And we can simply use the 2 target patterns as weights. If an input pattern looks like a letter L, but was 1 bit corrupted, the 2 weighted sums are calculated as 4 and 2. And because 4 is greater than 2, the winner-take-all layer should compute that Y1 is 1 and Y2 is 0, indicating that the input is recognized as L. Now, in this example, the weights are binary, and the function is equivalent to computing the smallest Hamming distance between input and weights. But the model also allows analog weights, which are more powerful than binary. For example, if the input pattern is heavily corrupted, it may result in a tie and thus cannot be classified. But if the weights have analog values that show which bits are more important than the others in each memory, then highly corrupted input patterns may still be successfully classified. Now, because of how simple the circuit function is, there exists a set of also very simple chemical reactions that can implement the winner-take-all computation. And these are the 5 types of reactions, and they can be classified into catalysis, conversion, and annihilation. So here I will explain 2 types of reactions, annihilation and catalysis. The annihilation reaction allows all molecular species, in our case DNA strands, to compete with each other until there is only 1 winner left. And then the catalysis reaction allows the remaining signal species to be amplified to a desired concentration. So the mechanism that we use to program molecular interactions is called DNA strand displacement. And initially, these 2 strands are bound to each other. Then an invading strand with the right sequence, including a short single-stranded domain called toehold, can bind to the partially bound strand. And then the 2 strands will compete for the complementary strand, and the previously bound strand will be released. So the invading strand can be seen as an input signal, the released strand an output signal, and the initially double-stranded molecule can be seen as a gate molecule that produces an output, but only when it receives an input. Now, this animation shows the dynamic process. The invading strand has a toehold, in this case colored in red, which binds to the complementary toehold domain hanging off from the double helix, starting a competition with a blue strand. So you see that the base pairs are being opened and closed very quickly, moving back and forth in a random walk process until the blue strand is released and the invading strand now becomes part of that double helix. Okay, so the annihilation reaction is facilitated by a double-stranded DNA molecule called annihilator. It can bind to a single-strand DNA signal Si. And because the DNA sequence of the orange domain in the signal molecule is designed to be identical to the orange domain in the annihilator, so they compete with each other for the complementary strand. Now, if only signal Si is present, this process is highly reversible, and the signal strand will not be consumed. But if signal Si and Sj are both present, Sj can bind to the annihilator from the opposite side, and then both signal strands will branch migrate to the middle point when the annihilator will be split apart, and both signal strands will be turning to waste product that cannot react with each other anymore. So this process ensures that different signal molecules will continue to kill each other until there is only 1 winner left. And then the signal strands, that is the concentration of the winner, can be restored through a catalysis reaction. The signal strand, now in this case named Sj here, reacts with a restoration gate to release an output strand while being bound to the gate bottom strand itself. At this point, the toehold on the right side of the gate becomes open, allowing a fuel strand to bind and displace the signal strand. So essentially, the free fuel strands are converted to free output strands and catalyzed by the signal strand Sj. So now when you look at these two types of reactions together, you see that it is important that the annihilation reactions between all pairs of signals take place first before the remaining signal is amplified. To accomplish that, we designed the annihilation reaction to be much faster than the catalysis reaction so that the signal species will preferably react with annihilators. And the difference in rate constant is achieved by having a longer toehold on the annihilator than that on the restoration gate. It is known that the rate of strand displacement grows exponentially faster with longer toeholds, which is a particularly useful property for tuning reaction kinetics within a range of 6 orders of magnitude. Okay, so similarly, all other reactions, all other functions involved in a winner-take-all neural network can be implemented using a set of also fairly similar and simple DNA molecules. And once we mix these molecules in a test tube, they can perform surprisingly complex tasks. So in our study, we chose a well-defined and understood task, handwritten digit recognition. But in real-world applications, these molecular handwriting could represent any information of interest, such as a gene expression profile, and the recognition results could represent any desired classification task, such as differentiating viral and bacterial infections. Now, the digit recognition task allows us to evaluate the complexity and diversity of molecular patterns that a DNA neural network is capable of recognizing. For example, the complexity will tell us how many unique microRNA or mRNA species can be simultaneously detected. And then the diversity will tell us how different an individual's gene expression profile can be tolerated compared to a common profile, for example. So we took 100 example 6s and 100 example 7s from the MNIST database, which is often used to test the machine learning algorithms, and then we averaged them to determine the weights. Now, these weights are analog. Darker bits indicate they're more commonly used in the examples. And each bit in the memories correspond to 2 DNA molecules that perform the weight multiplication function. And the concentration of these molecules corresponds to the value of the weights. When the neural network is used to recognize corrupted test patterns, each bit in the inputs correspond to the presence or absence of a specific DNA strand. So in the database, there are a total of over 14,000 example handwritten digits 6 and 7. If we look at where the example patterns are in the weighted sum space, this tells us how easy or hard the winner-take-all competition should be. Further away from the diagonal line is easier, and closer to the diagonal line is harder. So you see 2% of these patterns are on the wrong side of the line, which means it's impossible for our circuit to recognize these patterns correctly. 8% of the patterns are fairly close to the diagonal line, which we expect to be experimentally difficult. But 90% of the patterns are far enough from the diagonal line, which we expect our circuit to recognize correctly. So then we chose 36 examples from these patterns based on a uniform distribution for experimental demonstration. And the perfect patterns, which are the same as the memories, but in binary, were correctly recognized. The green fluorescent signal going high indicate a 6, and the yellow fluorescent signal going high indicate 7. But more importantly, patterns with up to 30 bits of deviation from the memory, which means very sloppy handwriting, were still recognized. And then to scale up the number of memories, we took a grouping approach and divided 9 memories into 3 groups in 2 ways, shown as 3 rows and 3 columns. And each Y output indicates which row the memory is, and each Z output indicates which column the memory is. And we determined the weights for each group using an average, then subtract method. Take the average of 100 examples of each in-group digit, subtract the average of 100 examples for each out-of-group digit. Of course, you can imagine that much more sophisticated learning algorithm could be applied to improve the performance of the neural network. But we intentionally chose a very simple one so that learning can later be implemented within the molecular system itself rather than being done on the laptop. And I'll come back to this later near the end of the talk. Okay, so our experimental data showed that indeed, all 9 digits can be simultaneously recognized for a total of 99 representative examples that we chose. And Kevin developed an online software tool. It converts a user-defined set of memories and test patterns into program code describing a neural network. which can then be used to simulate the kinetics of the DNA neural network's behavior. And it also provides sequences of DNA strands required to experimentally construct the DNA neural network. So this is what the web interface looks like. And we used this software tool in a class. Even undergrads and graduate students with minimal prior background were able to design and construct their own DNA neural networks with pretty complex behaviors. Okay, so I don't really spend time thinking about applications, but because I'm much more fascinated by fundamental architectures of programmable molecular machines. But I'm happy to tell you a few possible applications of DNA-based neural networks or DNA circuits in general that are related to work from other research labs. So the first example is molecular diagnostics. The CELIG lab at UW has shown in a proof of concept demonstration for differentiating viral and bacterial infections using a DNA-based linear classifier with 7 inputs. In principle, much more complex gene expression profiles involving at least dozens of mRNA transcripts could be classified using DNA-based neural networks. But one of the main challenges in these applications is the low concentration of RNA in blood samples and the high concentration of input signals required for the DNA circuits. Thus, developing a fast and ultra-sensitive DNA-based signal amplifier is an important step towards successful applications of DNA circuits in molecular diagnostics. As for therapeutics, the challenge includes scaling up the number of DNA circuit components that a DNA robot can incorporate and improving the robustness of strand displacement reactions within a biological environment. The next example is cell type imaging. The Stoyanovich lab at Columbia has shown that simple strand displacement circuits can be used to label B cells and T cells with distinct DNA barcodes based on the unique combinations of cell surface markers. And different from the well-mixed DNA circuits that I talked about earlier, the circuit components here are localized on cell surfaces and interact with each other through diffusible membrane proteins. Because many aspects of the structural and dynamic properties of the membrane proteins remain unknown, a challenge here is to understand the engineering principles of localized DNA circuits that exploit spatial information for computation while being robust to varying distributions of geometry and diffusion kinetics. And after the cells are classified and labeled with distinct DNA barcodes, the barcodes could then become the target for localized signal amplification, allowing unique cell types to be imaged with unique colors. Besides cell type classification based on surface protein markers, it could also be based on mRNA profiles. So HCR, which is a hybridization chain reaction, is widely used method for multiplexed mRNA imaging developed by the Pierce lab at Caltech. It is conceivable that these imaging techniques could be combined with DNA-based neural networks to embed molecular information processing within imaging. Okay, last example is water pollution detection. So the Lux lab at Northwestern has shown that an in vitro transcription system can be freeze-dried and distributed for in-field detection of water contaminants, including antibiotics, small molecules, and metals. And the system can be composed together with DNA strand displacement circuits for more sophisticated information processing. So it is conceivable that DNA-based neural networks can be used here for in-field classification of water samples based on complex profiles of contaminants. And similar to in-field disease diagnostics, challenges here also involve speed, sensitivity, and specificity. Okay, now let's return to the fundamental questions that motivated my research. Some objects are currently classified as matter and others as life. However, it is hard to define a strict line between them. More likely, there is a gap between the two. And once filled, the spectrum will be continuous. At that point, we will be able to explain how life arises from a collection of lifeless molecules and to engineer various forms of matter with desired mixtures of lifelike and non-lifelike properties. To explore the space between matter and life, let us begin with their similarities and differences. So biology provides ample examples of life forms for 4 billion years of evolution on Earth. Each of them can be seen as a molecular program, where information encoded in molecules determine how they perform a task. This is not unlike how information encoded in silicon determines how a computer program runs. Now, the kinds of tasks matter can perform, also known as a design space, depends on the nature of its substrate. When the technology is mature, the design space can be impressively rich. Over 1.3 billion lines of code in computer programs are produced every day. Presumably, the design space of possible molecular programs is vastly larger than the space occupied by existing examples. And synthetic biology has allowed some exploration of this design space, but our understanding of molecular programs is far less complete than computer programs. Yet, matter built from biomolecules will allow for capabilities more powerful than silicon-based computers, such as to replicate, grow, heal, learn, adapt, and evolve. Engineering this new type of matter, which we could call it programmable molecular machines, is key to closing the gap between matter and life. Now that chemistry has advanced to the point that biomolecules such as nucleic acids and peptides with custom sequences can be synthesized, the substrate of life has become available as a substrate for programmable matter. And to build something as complex as modern life, engineering concepts that led to modern computers must be transferred to molecular programs, including formal programming languages that define which instructions are valid and how they can be composed. But architectures for molecular programs must be fundamentally different from computer programs, respecting and exploiting the nature of biomolecules. So to systematically develop a molecular programming language and its supporting architectures and algorithms, a 30-year challenge is to build a synthetic cell. That is a synthetic molecular system of complexity similar to a biological cell. By building the system, we will be able to learn how to design structural, mechanical, and computational components that are fully composable and programmable, to store and consume chemical energy over repeated cycles, and to demonstrate sophisticated lifelike behaviors, for example, including learning and self-improvement. Here I show some examples of research directions. Each of these focus on several main principles governing an activity central to life. So things I chose to talk about DNA-based neural networks in the earlier part of this talk. So let me talk a little bit about related challenges in engineering molecular systems with learning capabilities. Learning allows life to be born simple, but developed into endless diversity and richness. State-of-the-art molecular circuits, once built, they have predetermined function. So to create a synthetic cell capable of becoming smarter over its lifetime, two general principles should be investigated. First, in nucleic acid or protein circuits performing neural network computation, adaptive memories can be developed where molecules encoding a memory can be selectively inhibited or activated. So at the principle level, this would be similar to the change of synaptic strands between biological neurons. Then with adaptive memories, it will become possible to engineer molecular circuits with self-improvement capabilities. For example, input signals will trigger a response of the molecular circuits to adjust their memories, which improves their capability for recognizing and acting on similar molecular patterns in the future. So there are three general classes of learning behaviors. In supervised learning, the circuits will be exposed to examples of what they may encounter in the future and examples of desired response. In unsupervised learning, the circuits will be exposed to only what they may encounter, but not what they should respond. And then this information alone will be used to improve their capability for handling similar situations in the future. And in reinforcement learning, the circuits will initially receive the same information as in unsupervised learning, but then they use the information to take an action, which triggers the reward or punishment in the environment, feeding back to the circuits to influence their future actions. So to build memories that are adaptive, the concentrations of the weight species encoding the memories needs to be adjustable in response to molecular signals. So for example, the weight molecules can be designed to be initially inactive, and an appropriate collection of them will become activated in a training process. For example, in supervised learning, a set of training signals and a label strand will be simultaneously present, and the label strand will indicate which class the training signals belong to. While in unsupervised learning, no label information will be present. So the neural network must compute which class a set of training signal more likely belongs to, and then activate an appropriate set of weights accordingly. So a critical requirement here is that the molecular circuit must be capable of multiple rounds of computation, generating different outputs in response to different inputs. But the current DNA neural networks that I talked to you today cannot do that, because the molecules composing the network, such as weight molecules, gets used up after a single round of computation. And thus the challenge of learning is closely tied to the challenge of metabolism. We need chemical batteries that can release fuel molecules to power each cycle of computation, or molecular circuits that can be recharged by a common form of energy, such as light or heat. And in fact, all of these example challenges are interconnected one way or another, which can only be accomplished by a strongly connected and intellectually stimulating research community. So the first synthetic DNA nanostructure was created by a chemist, Nat Seaman, in 1983. The first synthetic DNA computing device was created by a computer scientist, Len Edelman, in 1994. Since then, many more synthetic structures and devices made of DNA, RNA, and proteins have emerged, exploiting increased understanding of these molecules' thermodynamic, kinetic, and mechanical properties. Chemistry and computer science perspectives have grown strongly connected into one grand vision. Information-bearing biomolecules provide the ideal substrate for a new programmable matter, which will eventually enable powerful and embedded control within chemistry, medicine, and materials. Okay, so I'll end here. I feel very lucky to be part of this community and to have worked with a group of extraordinary students and postdocs who are excited about the future of programmable molecular machines. And I hope we'll soon be able to stand next to each other again. By the way, Greg Tigomirov, the tallest person in this picture, is also a Foresight Fellow. He's starting as an assistant professor at Berkeley, and he has open positions for graduate students and postdocs. So please contact him if you're interested. He's a wonderful scientist and a wonderful person. I'm sure you will enjoy working with him.